Yeah, so thank you so much for having me. I'm really excited to be here, getting to talk to a lot of people in soft matter from a community that I've read a lot about, but haven't necessarily seen in person. So today I'm excited to share some of the work we're doing in our group, which is all about mechanical forces at biological interfaces. And even though these are quite different sizes, from cells all the way up to the human sense of touch, what these all have in common is that these are mechanical interfaces. These all kind of obey continuum mechanics. So if we get good at understanding this system, maybe we can use some of those tools here and vice versa. So first things first is seeing the slide work. So that would be the first thing. Yeah, fantastic. Thank you. So where is Delaware? So Delaware is where we're at, and we are, if New York's up here and Washington, D.C. is down there, we're roughly in the middle. So we traditionally have been there for 25 years, and we have a lot of soft matter people. None of this stuff will necessarily make sense unless you are in the US system. But if anyone has questions about what the US system might be like, I'd be happy to answer those. But in our department, we roughly have two areas of materials categorized by their Young's modulus. So we have people working on the hard materials and people working on the soft materials. And so we have a lot of different people kind of working from synthesis to characterization to from structural polymers to more advanced biopolymers, organic electronics, and all the way up to mechanics, which would be me. So we have a big range, and it's a good place for soft matter. But so since there's so many people here who have done soft matter, I'm going to skip my intro slides, which is normally why do we even care about soft matter? And I'm going to be talking about two places where we care about soft matter. And so soft matter is all about it's the fact that it's deformable, makes it much more complex, but also more interesting. So we work on two different scales. One is, I think, more traditional disease modeling, but the other one is the human sense of touch. And so roughly sizes at the bottom. But today I'll first start by talking about the human sense of touch. So if you think about touch, today you got up, you got dressed, you felt your clothes, you felt the floor around you, you got your phone, probably swiped through some things. So you've used your sense of touch many times today. And what's interesting about touch is that compared to our other senses, we're very bad at controlling the sense of touch. So think about this television screen, which you are seeing. Think about a surround sound system for your sense of hearing. And these are very good at recreating those sensations. But for touch, I think we're much worse. If you think about the everyday technology to simulate touch, it's probably your phone, which vibrates. So a really big difference between what you can feel something like this versus what your phone can do. So we're so good at our other senses. Why is touch so hard? I guess to even answer that, why is touch even important? So one of the ways we're really interested in tactile technologies is to work with people with low vision or blindness. So if you have low vision or blindness and you want to, for example, get around this building here, you could typically pull the map if you're sighted and it's on your phone and you can see a map. But if you can't use your sense of touch so if you can't see a map, then you can start to make a map like this, right? This is a tactile version of a map in which you can have different floors and you can have stairs and you can have the bathroom over here. But this is enormous, right? In order to get this spatial layout, you need a very large device, which is at the center of a lot of discrepancy between socioeconomic outcomes, meaning like why are jobs for people with low vision and blindness typically not as high paying? Thinking about the field of engineering and physical sciences, how much we use visual aids. So for example, down here is a plot, right? So if we want to overlay, let's say, I ran my experiment in 5 conditions, like 1 molar, 2 molar, 2.5, and I want to plot all that data on top of each other, right? This is something you might do every day. But if you were to do a tactile version of it, it would be very difficult. The reason why is that this line is solid, and then the next one might be dashed. But then what should my third line be? Should I do solid dash dash, or maybe dots, smaller dots? This is all very difficult to interpret, especially as you lay them on top of one another. So there's this fundamental limitation on the types of tactile sensations we can create. There's also very, I think, straightforward applications of tactile technology. So if you think about virtual reality or maybe doing remote surgery, or if you're thinking about kind of the other way where you're trying to create a prosthetic, how can I better recreate tactile sensations? So because we're in a physical science area, but touch can seem as if it's very subjective, there's a lot of different aspects to touch, and it involves a lot of different fields. So taking you from the outside world in, you can imagine you're touching an object. That object can have material properties, physical properties, coefficients of friction. Then you have this object and then you touch it. And if you think about how you're touching an object, then you move with a certain finger velocity, you move with a certain finger pressure, and your finger itself has multiple properties. It's a multi-layered structure. It's heterogeneous on the surface. You have a bone underneath. Once you generate those mechanical forces, that's transduced into your body through mechanoreceptors. So these are mechanically sensitive neurons that transduce mechanical stimulation into electrical signals. And all this is through different mechanoreceptors. And all those signals are pre-filtered and interact in interesting ways, but ultimately go to your brain to form a single stimulation in your brain. And you make a judgment on that, right? So you may have a certain perception with that. So between all these different fields, where does soft matter come in? One thing I really think is important is that if you think about the actual forces, this is how you decide how something feels. It's all the forces at that interface, which I think is a very traditional field in soft matter, right? Contact, friction. So this is where I think there's a lot of opportunity from soft matter. So if you look at the finger itself, this is a traditional soft matter problem. It's deformable. You have fingerprints, meaning it's patterned, and those will change the forces. How you touch an object matters, right? So if you slide versus you tap, you have a different mode, right? So mechanical testing, we know those can give very different outcomes. And you have a different propensity to use different forces and different velocities as you explore, which will change the regimes in which you're touching in, will change the forces you get. So in touch, people typically do two types of studies, or at least the way I organize it. So one is maybe new technologies to create touch. So imagining I want to make a tactile display, I want things to feel different. So maybe I can have some pins move up and down. And the other one is studying the sense of touch. So how does this fundamentally work? When I say something is soft, how do I know that someone else is saying the same thing? So in both of these, we ask how might soft matter mechanics and materials benefit touch? And I think the most important aspect of this is that we want to be deterministic. So something we're trying to move away from is just purely data-driven, right? I could just go to some hardware store and buy a million different pieces of things and touch them and then organize them. But instead of that, we're trying to maybe have some hypothesis-driven work. So if we think about ways to generate touch, you can think about, well, what's the first thing you might think about? You might think about Braille. And even though this is a relatively new material, this is very old technology, right? You could carve out wood and make little bumps on them for a very long time. You could make these bumps go up and down, maybe with an electromechanical array. And these are the typical ways you might imagine creating touch, right, in a synthetic device. So as people were looking to improve on this technology, they might ask themselves, well, what's the smallest little bump I can feel? What's the smallest thing I can feel? So you can do this through filaments of ever smaller and smaller sizes, so they have lower and lower bending rigidities. And then you say, can you feel it? And then on your fingertip, maybe you can feel them quite close. And on your back, they would feel quite far apart. Also, you can use maybe more modern techniques to create a bunch of different buckled films with different amplitudes and different roughnesses, which people can actually tell apart with a 13 nanometer difference in the wavelength. So if you have a bunch of arrays, their amplitude changes by 13 nanometers. Skadung et al. Show that you can tell them apart. But what all these things have in common that I think represents a place where there's a lot of opportunity is that all of these are physical technologies, right? These are all based on making a bump, a geometric feature, and colliding with them. But knowing from adhesion background, not only does the physical layout of the interface matter, but also the chemical composition. So the way we're starting to look at this is, well, what is the smallest thing you can feel, but maybe from a chemical perspective or an interfaces perspective? So if you think about that, if I want to establish this, I need to create a system that has very minimal physical differences. I need a way of controlling the surface chemistry. And then from there, maybe I can start to connect changes in surface chemistry with the human sense of touch. So if you ask for a system like this, I think most of us as physical scientists and engineers would come up with this system right here. Self-assembled monolayers. If you've been in a clean room, you've done this many times. And these are silicon wafers, which are mass produced to be very smooth. And self-assembled monolayers have a chemistry which preferentially allows for functionalization in an ideal world, right? That it's more preferential to form this monolayer with a different functional group. So what I can do is buy this array of self-assembled monolayers, and I can try and be methodical about this, right? I can try to slowly increase the chain length. Maybe I can throw in a nitrogen in there. And then I can put them on top of my self-assembled monolayers. And if successful, we should be able to find certain pairs of this that people can tell apart by touch. So the way we do this is called an odd man out test. So a human subject, you give them 3 things. 2 of them are the same, and 1 of them is different. So you have to say, okay, it's the middle one that's different. So this is kind of where we're at when we first started, right? We have this list of materials. We know how we want to show it's true, but how do we get from this long list to something more reasonable here, right? We know the chemical structure. What are people actually going to do? So because I did not want to test 36 possible combinations with like 100 people and spend an entire year doing this, we need some way to narrow down this list of materials to something much more reasonable. So the first thing we did is, well, what does a person do when they actually touch this surface? So what they do is they slide their finger. You can see this here. There's one wafer on one side and another one on the other. And right there, that's kind of our running hypothesis, right? So the mechanical forces that give rise to tactile stimuli. If we understand the mechanical forces on the finger, we may be able to predict what a human's going to do. So we can do something like that, right? We can build an apparatus that is quite simple, but in some ways mimics the properties of a human finger. It won't be perfect, but we go after some of these properties, such as its kind of its interface. And we can test this at different masses and velocities. And once you do this, you can get maybe something like this. So in red is the friction force from one surface, and in blue is the friction force from another surface. So even though we can test this, now we have a new problem. And we have 2 types of problems. And one is that no one knows what to do with any of this data. So this curve here, like, does that mean it's good? Is that bad? Is this different? Is this not? So that was actually a big problem. And the second one, which is actually also huge, is that people change how quickly they go. Maybe in the lab, your machine can go at a certain mass and a certain velocity. But when you're doing this, I promise you're not going at the same exact velocity. So how are we going to connect all these 2 challenges together in order to get something useful from this and hopefully predict what our materials will do? And so the traditional way of looking at friction and touch is to do this, right? I have a bunch of materials here, and I'm just going to get their friction coefficient, right? So looking back at maybe, I don't know what the equivalent of high school physics here would be, but looking back at the first time you've been exposed to physics, okay, each material has a friction coefficient, and I times it by the normal force, and that's my friction force. So if I do that, I could do it for nylon, and for some given force, I get, this is my friction force for nylon. And then I can find another material like glass, which has a higher friction coefficient. And then for the same amount of pressure, then I would get, a higher force. And maybe I could say, these 2 materials feel different because they have a different friction force. But if I just look at this, then this means if I take the nylon and press just a little harder, I should get the same force as glass or even higher than glass. So from this framework, anything you touch, you just press harder, it'll feel like everything else, which clearly isn't true. So where is the information missing? So if you look at a real friction trace, and you look very closely, there's a lot of oscillations. These oscillations aren't just noise from your sensor, like something's wrong with it, but these are actually real mechanical events caused by stick-slip friction. So multiple different events happening at the nano, microscale, all coming together in a macroscopic object. So what we think is that it's the information here that's really important for telling surfaces apart. And the analogy I'll draw is that how do you think people tell 2 songs apart? It's not by the average volume, but rather by the harmony and the notes in between. So we started to say, well, maybe this would be the important information. So we have that, which is great. We also have this issue that humans vary velocity and mass. And if you've done any mechanical testing, then you know this can have profound effects on your results. So here again is that same friction and velocity where things look a little different. So maybe it's these wiggles here that I care about, and that's why I can tell red from blue. But then if I change the velocity and mass to a very reasonable velocity and a very reasonable applied pressure, then to me, these look the same. So now I don't know which one of these is a human doing. Are they using this or are they using this? Both of these are reasonable for humans to do. So how can I reconcile the two?
Zero. So if there is zero, there is no contact.
No, I'm sorry. It's applied mass on top. So it would be the dead weight of the finger. So zero is how much the finger itself weighs without any additional mass. So because of this, I have one situation where they look different, another situation where they look the same. We have to change our way of thinking from how can we tell these two surfaces apart to what conditions, when am I going to be able to tell this apart? So in order to do that in a more efficient way, based on this idea that mechanical forces are tactile stimuli, what we did is we just quantified the total differences between these traces. So on the left, we have friction traces on one surface. On the right, we have friction traces on this surface. And then we do a thing called cross-correlation, which is a very agnostic way, meaning I'm not doing anything suggesting of the underlying mechanisms. I am just blanketly saying how different or how similar are these two. And so what I can do is I can do a cross-correlation between these, and then I get an idea. Okay, this would be how similar and how different are my two surfaces. So meaning I can do a cross-correlation under one pair here, and you can see that it's very large and symmetric. So I'm going to score that red because actually those friction traces are very, very similar. Now I can compare that to maybe something else where the friction traces are giving a cross correlation that's very low and asymmetric. And so I'm going to score that green because the friction traces that led to this situation means that my two surfaces are very different. So now I'm going from friction traces into a very simple single outcome, red or green. So what I can do is now take all my materials and across my different velocities and across my different masses is construct what we call a discriminability matrix where, okay, for this material, there's maybe only one condition where the friction traces are different. So I don't think people can tell this apart easily. Versus I can think of other combinations where we do see a lot more green. So there's more chances of where the friction traces are different. And maybe when someone explores the surface, they're more likely to hone into this. And the great part of this is that once I've taken my mechanical testing on each one of these surfaces, that's it. I can just compare it to my existing library. If I were to add another material, I don't have to redo this entire thing, which is much more sustainable. So of course, we have to actually see if this works. Do humans have anything to do with our mechanical test? So again, what we do is an odd man out test where two of the surfaces are the same. One of them is different. The one that's different is randomized and put in a different location and so on and so forth. But what we see is that, so if we have these two surfaces, which have a very similar chemical structure, except that the carbon is 1 unit longer, people can't tell that apart. And when we do this internal nitrogen substitution, people cannot tell it apart. But if we make the change in the carbon much longer, so if we increase the length by 3 carbon units, people can actually tell this apart by touch. So again, these are on top of silicon wafers. They aren't that physically different. It's just really that surface chemistry that's different. And so if we do this and we change the terminal carbon for a nitrogen group, we also see that people can tell this apart. And if we include this effect of increased chain length and this carbon for nitrogen substitution, people can tell this apart with the greatest accuracy. And so if we see this, then we can say, well, did this actually correlate to some of our mechanical testing? I would say yes, somewhat we got a hint from our mechanical testing that this would be different and this would not be different. So it's our way of screening through mechanical testing what humans might do. And also this helps us find that, maybe some of these subtle effects, the reason why touch is so hard to recreate is because we don't have a lot of new ways to recreate touch. So maybe by using surface chemistry, it opens up a much wider avenue of different ways to stimulate the sense of touch. And so really we don't have individual receptors on our skin like the way you have for smell or touch that can recognize chemical functionalities. So really what's happening here is that if you have a long enough chain length or enough intermolecular forces, basically it goes from a liquid-like surface to a more solid-like surface. And so people have known this from AFM that you will have much higher friction at lower chain lengths. And this has been shown through AFM, which is exactly kind of what we see. So overall, I wouldn't say that it's a 3-carbon difference in length that really leads to this ability to tell surfaces apart. It's more the idea that you can use differences in monolayer ordering to create tactile contrast. So however you achieve that, whether by getting longer carbons or substituting in nitrogens for carbon, will lead to the same effect, which is what we see. So with that, you can kind of see how we've built towards this kind of materials library construction, right? We have all these materials. If we want to test another material, we don't want to redo all this work. We can just add this to our database. And this would be our running prediction, right? So we didn't test on all of these. These are the conditions which we did human testing on. And then we fit the results to the human testing. And so we would predict for the ones we didn't test that, okay, people can't tell this apart, but they can tell this apart. So whether all of this is true, we would actually have to test. But I think this is the idea that we're going towards. So what can we actually use this for? So again, I said that a lot of the work we're doing was motivated by helping people with low vision or blindness. So imagine this is a microwave sticker. So a lot of times microwave symbols for start and stop and for heat are not, you can't feel them. So if you couldn't see them, then you wouldn't know which button did what. So then you need these stickers to say go or stop and so forth. So what we did is we saw if, so this is our results from earlier. And we see that people with low vision or blindness can also tell these surfaces apart. Their success rates are different. But what that means is we think that some of these chemistries could also be used in this application. So we had a game we played. And in this game, we took cards and we coded all the red cards with one type of coding. And we took the black cards and coded them with a different type of coding. And then we randomized them, we trained subjects, and then we gave 10 random ones in a row. And they said, is it red or is it black? So just by touch from this coding, they had to sort. And we found that, and this is all blind and braille literate subjects, so maybe the most tactile proficient subjects, that some people got these 10 out of 10. And one subject was even able to self-correct, meaning that they put it in one category, then they touched again, said, I'm not sure, and put it back in the right category. So there's a lot of questions I think we can go on from here, but just being able to use this immediately, I think, in an application for creating maybe easier types of signage or many other applications in tactile aids. So kind of going back to the materials discovery, I think what we've shown is kind of a pipeline. How would you actually then discover new materials and use this for a broad range of phenomena? So earlier we found that monolayer ordering is a source of tactile contrast, but now we can start to apply this to polymers, right? If you think about long-lasting durable materials, polymers fit the bill. So here we've created different surfaces with the same chemical formula, but different levels of crystallinity in the polymer. So one way is through annealing at different temperatures, and another way is by varying the tacticity of the starting polymer. So this isotactic one is more likely to form crystalline domains. Depending on the audience, so this is like, right, the same material, but through different annealing, you can get very different structures. So we can measure all this, right? So we have 4 different polymers, all have, again, a similar formula, but different levels of crystallinity. We measure the crystallinity through wax. And basically, so I have 4 materials with 4 different levels of crystallinity. And so we can see, can people tell the level of crystallinity apart? And so we have this workflow, right? So we make our samples, we characterize them through traditional ways. We do our mechanical testing to give us an idea of which ones may feel different. So in this case, my list was pretty small, so I probably could have tested them all. But right, if I want to test 10 materials, then this will become critical. Then we compare our friction traces and basically do our human subject testing. And here we start to see that people can tell surfaces apart even by their degree of crystallinity. So hopefully what you're seeing here is a new way of just screening through all these very well-known microstructural changes and seeing if they're actually useful to create new types of tactile sensations. One thing we are working towards, so let me pause. One thing we're working towards is this step here is, this is the best we knew at the time. We didn't really know what parts of this friction trace were important or not. So we just said how similar or different they are. But bring this back maybe closer to soft matter mechanics. how can we maybe connect some of these wiggles, right, to something a little bit more into what people actually do? So the origin of these wiggles, right, are mechanical instabilities between sliding, elasticity, and adhesion. So one of the things we started to look at is, well, maybe can we create a phase diagram based on this? So here's a stiction spike, and here's no stiction spike. These we call a slow frictional wave, and this is the lack of a slow frictional wave. So we just tried to find whether these instabilities occur or not. So doing that, we can take our material and categorize them by where these instabilities occur. Sorry for this butchered figure, but I'm just trying to make it easier to see. So the color represents the type of friction feature. And right now I'm only showing circles. So for APS, you can see that there's this transition between the presence of a stiction spike. Here, there's no stiction spikes. And here, the presence of slow frictional waves. So what I can do is I can look at my other materials and map out where these actual instabilities and transitions in instabilities occur. So what you can see, for example, here is that this circle and this triangle are a different color. So these two materials are experiencing a different type of instability under this condition. So if we do this, what we can do is now, well, why don't we just count up the number of times the instabilities are different? And when we do that, we get pretty good agreement with human results. So what we're saying is that these instabilities occur at slightly different velocities and masses due to the adhesion differences between these materials. And maybe that's what people are looking for. Maybe this is how people can actually tell surfaces apart. So taking these friction traces and trying to find what are the salient, what's the important part of this friction trace? So this is definitely work in progress. So what we've done is gone back and kind of analyzed all of our data, which takes some time, and trying to create these categories of steady sliding, stiction spike, slow frictional wave. And of course, these have deeper kind of underlying mechanics reasons for why we're picking them, which is what makes it more attractive to us. And we see some correlation. So we're going back and trying to test this hypothesis more rigorously. But this would be great because I think it's been very hard to say, well, why do two objects feel different? And, we can't really pinpoint it to a chemical structure because we can change the morphology. It's clearly not just the friction coefficient. But this would start to give a little bit more fundamental reason of why can we tell objects apart? But we'll see. So it's the hypothesis, and that's very much active area of research. So to kind of conclude this part, we're taking this kind of, if you think about touch as being a very physical-based construction in which all these objects are poking, maybe bumps on a surface. And what we're saying is, can we start to bring in surface chemistry to widen the range of tactile sensations we can create? What would that kind of look like? And why would we do this, right? So as soon as we start to bring in the chemistry to it, you can do all sorts of transformations that would normally belong in the chemical realm. For example, an antigen detection that then transduces to a tactile thing. You can imagine doing something that changes with light, maybe something that changes with temperature, changes its morphology, and maybe that will also change its tactile sensation. So thinking of dynamic systems. Ideally, we're also trying to answer fundamentally, how do humans perceive touch, right? As soon as we want to do any work in this field, I feel like we jut up right against the neuroscience, right? What is it that people are feeling? So that's kind of the first part on how we create touch and trying to use soft matter mechanics to create touch. The other part is also how we study touch. So how does touch work? What does it mean when we're talking about a tactile perception? So one of the things that I think has been very useful for vision is that people know that red, green, and blue stimuli can be recombined to create a wide range of colors, right? And so from this fundamental basis of red, green, and blue, you can recreate a lot of different stimuli. There's nothing like that for touch, right? We don't know that if you get a hot plate, you get sandpaper, you get some sticky goo, I can somehow combine this in some way to recreate any kind of tactile sensation. But this would be very useful, right? So this is how they design TV screens, right? As long as I have enough red pixels, green pixels, and blue pixels, and I know the intensity of them, then I can recreate it. But again, nothing like this, right? And that's the really hard part, right? Unless I brought my samples today, you wouldn't really know what my samples felt like. And so I can't even communicate what they feel like, and which is a really fundamental barrier to touch research, right? Whereas I can give you a color, a Pantone, a hex code, and you would know what color I'm talking about. So where we started was, well, how do you perceive softness? An object that is soft. So you could think it's a material property, right? This is rubber, and that's kind of squishy. And this is steel, and that is not very squishy. So maybe it's a material property. But what about a feather? A feather is made from keratin, it is a very hard protein, but I think most people would say it's soft, right? So there's obviously some geometric component to this. And so I think some people in the field have kind of reconciled this in terms of protein contact mechanics, right? So, and this says that if I press into an object, I have a contact area across my finger, and the object goes into my finger by a certain depth, the indentation depth. And so this is great and all, right? Contact area, indentation depth. And so some people have known this, but the problem with this is if you work out the math, the relationship between indentation depth and contact area does not depend on the modulus. What that means is if I'm getting a bunch of different samples and testing them on people, I will never know if what part of that was due to the indentation depth and what part of that was due to the contact area. And that's the big problem. And R is the finger, and people are using the same finger. So what we did is we did two ways to break this relationship. One is very simple, where we just have some micro patterning, right? If you can't touch the bottom, then that reduces the contact area. And of course, it has to be small enough and so on and so forth. The other thing is this thin film rigidity. If you make a very thin film, then it appears to be much stiffer than its bulk counterpart because, right, you're building up that gradient at a much smaller thickness. The significant part of this for us is that now you have this thickness dependent, H, relationship between indentation depth and contact area. So using these two tricks, we can create a lot of different samples that vary in their thickness, their Young's modulus, and their effective surface area. And now we can finally start to tease apart what is the role of indentation depth and contact area on the perception of softness. So we do two types of tests. One is, this is called two alternative force choice. And so I give you two and you say, which one is softer, right? So like if you're getting glasses that are 1 or 2, this is exactly the same. And the other one is magnitude estimation, meaning on a scale of 1 to 10, rate your favorite. So on this one, and I'll walk through some of these. So this is the aggregate percentage, meaning I have one sample, it was dark blue. How many times was the judge softer than the other samples? So what can we tell? So I have the color as the Young's modulus. So the red is different, green is different. But anyways, so these all have the same Young's modulus. All my blue samples have the same Young's modulus. And even though they have the same Young's modulus, they vary widely in terms of their relative softness, right? So all I've done is I've changed the thickness. So we already know that this thickness is quite important. And so you can imagine it's really not the Young's modulus. This was the first thing I think we had to establish. I don't think it's a Young's modulus coming from a mechanics perspective, but other people working in the field, they might just think that softness is only the Young's modulus. So this one, I think we had to hammer home. The other thing is that you can also have this surface area and thickness dependence. But I think this is probably the most important comparison since there's too many to make. In this case, these are about the same, right? They have a pretty close thing. They are different in every single way. They are different in their Young's modulus, they are different in their thickness, and they're different in their micro patterning on the surface. But somehow, these all work out together to give about the same perception of softness. So beyond that, I think the most important finding we had at all was this is the aggregate data, but on the right, this is the raw data. So this is when someone tested dark blue, and did you think it was softer than red? Then I would have recorded the result here. And so this is all my head-to-head data of every single sample versus each other. This is reflected around this line, which you don't have to look at, but so just up here is all my raw data from all my samples. And the most important part of this plot is that there's no red above this line, and there's no blue below this line. And that's not some feature of the way this is plotted. That's something about my data. And what that's saying about my data is that if I gave someone sample A, and they found A to be softer than B, and then they found another sample, B was softer than C, then A has to be softer than C. And this is happening across 9 samples across 10, 15 subjects. So the idea is the order of softness between subjects and in between themselves is consistent. So this is called transitivity, right? A softer than B, B softer than C, A softer than C. That means that everyone has a similar concept of softness from the people we tested. That means if you're designing something for softness, it's possible, right? Before that, it could have been purely subjective. But now, if I design it and someone else tests it, or they test it on another day, there might be some reliability in their performance. So then, of course, we can do different types of fits, like, is it contact area plus indentation depth, or is it contact area squared? We did something funny, which was that we reran all our numbers as if your finger was very rigid, and that it was actually much better statistically. Obviously not true physically, but the human data was better explained, suggesting that humans ignore the deformability of their own fingers. But anyways, I think this is what people want, right? This is the equation for softness. So if I want to make something feel twice as soft and I want to change the indentation depth and the contact area, this is how they add together. So to make it more accessible, let's say I have 2 blocks and I want block 1 to feel twice as soft as this block. I need to reduce the Young's modulus by 1/3. So that's our finding. That's what you could use this equation for. If I want to make something precisely feel that much softer than another object. Okay, so I'm almost running out of time. So that's our human touch side. But now with the last little bit, I'll talk a little bit more on our disease modeling side, which is really talking again about these interfaces which are deformable and accessing them. So on this side, I would say the mechanical properties of small things are very important, right? Particles versus hollow particles, colloidal interactions depend on all this. But also in medicine or in health, a lot of diseases can be identified by the fact that they have different mechanical properties than the healthy version. So if you think about cancer and circulating tumor cells, which tend to be much softer than the cells around them because they can metastasize. If I think about fibrosis or scar formation, where I have cells that are much stiffer than the cells around them. So these mechanical properties can be very indicative of disease. So people know that mechanical properties are important in biology, but I think if you look at this, there's a lot of opportunity to develop new technologies which can actually access these properties. So if we look very small, there's atomic force microscopy. And if we look very large, then you can use your traditional tools. But as I'm sure a lot of people here at this institute can relate, there's this very challenging range of sizes that make it really hard to access these properties. And this is a size that's very relevant for organ on a chip, organoid, disease modeling. And so we think there's a lot of opportunity there. So we work on 2 different diseases. One is osteoarthritis, which I won't have time to talk about today, but the other one is this idea of fibrosis and scar formation. And again, this is really, we're trying to model a layer of cells, which really falls in this kind of range where there are not a lot of good technologies to measure this reliably. So the way we do this is through a method called which relies on elastohydrodynamics. So if you have a tube and that tube is soft and you flow water through it, the water will push out on that tube. And so that fluid-induced deformation is called elastohydrodynamics. And so what's good about this technique is that really the type of deformation you get depends on what's inside, right? So if the deformation will not only have an amount, but a profile. So whether I have a liquid, a gas, or a solid, I'll get a very distinctive profile. So I just need some way to measure it, and these are usually quite small. To measure that type of deformation, we put graphene very close to a channel. It's not inside, but it's very close. And so we use this to measure our deformations. So this is a piezoresistive sensor. So you're going to see voltage changes, but that's really just related to the deformation of my channel. So doing that, the first thing we have to do is actually verify we're measuring elastohydrodynamic phenomena. And so the classic way to do this is to make a bunch of different devices of different sizes and different moduli, and then change the flow rate, right? So I change the flow rate, it expands out more, so I can measure the deformation. And if I think it's elastohydrodynamics, then all of these should be able to collapse on a single plot. So, okay, I think the phenomena I'm measuring is elastohydrodynamics. So then we can do fun things like we can put a particle through here. So here on the right, you just saw some particles flowing in this direction very quickly. And then when I slow it down, we can get the deformation from a single particle going across this, right? So we haven't built that technology yet, but you can imagine if we could get this more sensitive, then maybe the mechanical properties of a single particle could be related to this kind of shape. This is the last way I'll show it, and this is the way we actually use this currently. So this is a bubble clearing through the channel. So it's filled with liquid, and then I shoot air through it. And so when that happens, you actually get a very strong signal on the sensor. And so what you're seeing here is this bubble transiting across the channel. So the bubble first enters the leading edge of the bubble. Then this is where the bubble is clearing out the channel. And you can see the squeeze film kind of there. And this is where the bubble has completely left. And because we have a voltage-based reading, it's really easy to get high frequency data. And you can see this relaxation where the wall has come out and it's going back in. So from here, we can take this and plot this against a simple spring and dashpot model and extract viscoelastic constants. And so from here, we get this kind of pretty close to literature values, I would say, where we just fit this very simple model to this kind of deformation profile. But the way we can really use this is that this reports on everything inside the channel. So what I can do is I can coat a second layer inside that channel, and it should change my response. So what we do is we take that microfluidic channel and we coat it with little discrete islands of collagen, and then we grow a monolayer of cells inside. And so working with the Zhao group, they grew some NDCK cells, which forms a monolayer. And so this is where the cells are seeded, and then they reach confluency by day 7, which you can see kind of that they're all nestled up against each other, forming a single layer. So what we can do is we can then flow a bubble inside. So we flow a bubble and you get the signal. And then this bit over here is the viscoelastic relaxation. So we can compare a bare channel to a channel with cells. And so when you do that, we can start to get viscoelastic constants of the actual cells themselves, right? So we just add that additional layer, and any change in signal should be coming from the cells. So what's great about this is all we need to do is flow a bubble. So what we can do is we can put in trypsin EDTA, basically dissolve the cells. And so this is what happens kind of visually, where you can see lots of individual cells here, and then over time, there's less cells. And so what we can do is just flow a bubble all throughout this, right? And start to see this decreasing, this increasing relaxation time. So on the right here, we have the cell count, right? Which I can just get from counting these. But on the left here, this is the actual viscoelastic constant of that layer in real time. So well, not quite real time. We still do have to flow that bubble. So maybe every 10, 15 minutes. But what this platform is now a way to directly assess the viscoelastic properties in a very convenient format, right? So I can take this whole device, move it into the incubator, take it out, move it over. I can do imaging at the same time because I don't need anything else in the fluid, right? I just need to flow a bubble. So we think that this could be very useful for trying to understand how maybe, if this was some disease where the modulus was going lower, maybe a drug introduced here would bring it back up, right? So it'd be a very good way to quickly assess these kind of in situ dynamics. So we think it's a good platform for that, but also we're really excited in its use as a way to investigate elastohydrodynamic phenomena, understand how these interfaces cause deformations, which has been kind of a explored area, maybe theoretically, but not too many experimental confirmations of these phenomena, which is, I think, very interesting. Yeah, so with that, hopefully you've seen kind of two stories about how mechanical forces at biological interfaces are informed by the underlying mechanics. So we used elastohydrodynamics in the last part. We're talking about soft sliding, friction, adhesion in the first part about touch. And these are all how we approach these problems, or we think about what's really driving these forces that we care about, and how can we get at them? How can we investigate them? And with that, I would like to thank all the people who have really worked on this. Tianzeng really led the work at the end. And Abby was kind of one of the first students who really helped drive the touch stuff forward. So with that, I can take any questions, and thank you so much for your time and attention.